mentioned, my name is Ekhtiari, and as the last talk that we have in this panel, I'm going to talk about ISAM Global Expert Network as a VCon. I work at the University of Minnesota, a Center for Neuropsychosis and Addiction, also Department of Psychiatry, and I do not have any conflict of interest other than fundings that I have from different organizations, different federal organizations in my works. And I basically put all my slides in the QR code. So if you want to have my slides, all the slides are already available on the QR code. And I will bring the kind of QR code at the end as well, if you want to have the slides. So then you can have all the slides they have here. So what is VCon? And what we try to kind of put together as ISAM Global Expert Network as VCon. VCon stands for Virtual Collaborative Networks in Science. And what we try to do, we try to promote diversity, equity, global representativeness, scientific rigor, open research consortiums, and joint decision making. There are some kind of global activities that people develop together, but they try to bring a group of friends and they try to put things together. But we try to make sure that there would be a global representativeness and kind of a significant emphasis on equity and diversity, and how we can bring all the people from different parts of the world in the joint decision-making process. But let me just give you an example of how we develop these virtual collaborative networks in science. And I'm going to talk about Ekhtiari as one of the VCons or virtual collaborative networks that we recently developed. So let's start with this slide. What is this? What is the name of that? Those who cannot kind of name the pastry here, they probably didn't have time to go around in Lisbon. And they spend the entire time in the conference center. So they didn't have time to go outside and find those places. What is the name of this pastry? Yeah, of course, those who are from Portugal, they know the answer. But those who are from outside, I highly recommend you to try them, especially when they are fresh out of oven. They are really, really good. So they are one of the best pastries I've ever experienced. And if I ask you in terms of how much do you like to have one of those really nice, tasteful pastries right now, what is the number from 0 to 100? 70, okay. And those who are from Portugal, they can tell us how is the quality of those kind of pastel de natas. Are they good? Are good. They are reasonably good. Okay. So there are different numbers. And this is what we call cue reactivity. How much we are responsive to a cue. And how much a cue could activate craving in us. And this is a phenomenon that we also have in addiction. We call addiction cue reactivity. We talk about how different cues could activate craving. Over the time, people kind of with different studies, different kind of methodologies, they realize that when we talk about drug cues and cue reactivity, they are important for the process of addiction recovery. And they are important part of relapse prediction. And there is a recent systematic review that is being published in Journal of Psychiatry concluding that assessment of craving and cue reactivity should be a part of routine clinical practice in addiction medicine, because the reactivity to cues and the craving that the cues could activate, that could be a good predictor of relapse. And that would be important to consider that as a part of routine clinical practice in addiction medicine. But cue reactivity could be kind of an important part of developing treatments for addiction treatment as well. So cue reactivity is one of the main cores that we have in addiction treatment, and how we can target cue reactivity to be able to develop different treatments. So it is important to be able to develop a virtual collaborative network around cue reactivity. And I'm going to give you an idea about how these networks could be developed and what are the different levels of collaborations that people can do through these kind of virtual collaborative networks. So we started with kind of the first level, and the first level is networking. So starting to see how we can involve in some communications and information exchange between different groups of people. And for being able to do that for the network level, that's kind of the first level of developing a virtual collaborative network. We started to share some ideas and exchange ideas through some webinars. And then we started to see how we can share some of the tools that we have with other labs. One of the tools that we recently shared in the network is what we call methamphetamine and opioid queue database. So this is a large database of 360 queues that could activate drug craving among those who are using meth and opioids. And that database is already available. And not only the drug queues, but also we developed basically controlled queues for those drugs, drug queues, to be able to use them in different settings, different experimental settings as well. And then we basically use that database and we developed a kind of brain responses database to those drug-related cues. And as you can imagine, when we expose people to those drug-related cues, those cues could activate different parts of the brain. We can have different neural circuits in the brain involved in response to those drug-related cues. And to be able to use that and expand that idea, we develop multiple tasks and multiple fMRI databases from those drug-related cues to be able to evaluate how different populations would respond to those drug cues. For example, we have a large database of a discovery sample, and we can evaluate specific brain responses to those drug-related cues. And we have replication samples in other clinical settings. And then we also have done some test-retest reliability assessments. And the good thing is that we have started to share all those databases with other labs that are interested to have those drug cues and also fMRI databases to see how brain would be responsive to those drug-related cues. So that was the first step in terms of doing some networking and sharing queued databases and also fMRI tasks that we are developing. And afterwards, different groups around the world, they start to do the same. So now we have multiple groups that are sharing their drug queue databases and also their fMRI tasks to be able to be used in other places around the world. But beyond networking, the next level would be what we call coordination. And what is coordination? When we kind of go to the next level, we try to align some of the activities to be able to have more efficient results. So how we might be able to align some of the activities that we are still doing independently in different labs around the world, how we might be able to make some alignments between activities that are happening in different labs. One of the things that we have started to realize that people are developing different queue-based interventions, but there is no consensus on how to categorize these queue-based interventions. So we organized a webinar and we tried to discuss about how to classify all those queue-based interventions and try to make a consensus around kind of intervention development based on the queues. And we are organizing those activities around a specific framework that we recently developed around attention, salience evaluation, interoception, control, and memory. And we are basically developing a new framework for consensus statements or consensus kind of around a categorization for queue-based intervention. The next level of coordination that we have recently started is about how to do an fMRI drug queue reactivity in the best possible way in different labs around the world. So we realized that we need to have a checklist and make sure that all the other labs that are doing fMRI drug queue reactivity, they will be able to follow the same kind of rules and activities, making sure that then reporting those activities in their final papers to be able to have a level of coordination across different labs in the world. So to be able to develop that methodological checklist, we started with a systematic review of all published studies with fMRI drug queue reactivity. And we have over 440 published studies with fMRI drug queue reactivity so far. So we made a steering committee of key figures in the field, and we brought people together, and we started the idea of what we called addiction queue reactivity, and we started a collaboration with Enigma Consortium, which is the larger fMRI consortium, which is the the largest fMRI consortium in the world. And we basically made a chapter within that Enigma consortium to be able to push this forward. So we developed the initial version of the checklist with a group of experts that we had, and then we revised the checklist, and then we invited all those people who have publications with fMRI drug cure activity in a systematic way, not just my friend and friend of my friend and friend of my friend, but also, we try to do it in a systematic way. So we try to kind of make a specific inclusion criteria based on a number of publications, and we systematically invited those people to make sure that everybody who's active in the field is invited to the party. And we try to kind of have their voices in the revision process of the checklist. And we revised the checklist over time, and we asked all those experts to rate how much all these items are important when we report a cure reactivity study. So we developed a checklist with 7 categories, 38 items, and 75 recommendations. And then we went back and started to, these are the major kind of items to be reported in an fMRI drug cure activity that we kind of made a consensus around. And we went back to the previous publications in the field to evaluate how much these factors are being reported in previous publications. What is the level of adherence to these sort of items that we think that are important to be reported in any fMRI drug cure activity study? And we showed that many of those items are really underreported in the previous publication. So we realized that there are, at least retrospectively, many kind of labs are not that much adherent to these checklists and these items. And if I show you the distribution, just minority of papers are kind of adherent to that checklist, and they are reporting things in a detail that you can basically replicate the study that they have. There are many studies that are published in the literature that you cannot replicate them because they have not provided the detail that you really need to know when you evaluate a fMRI drug cure activity study. And that is the way that we are encouraging any other lab which are going to do fMRI drug cure activity in the future to basically be adherent to that. And we are going to do this survey again in 10 years and see how much people kind of enhance their adherence to reporting the standards to make sure that the studies are being published, people can replicate them in the future. And we ended up publishing that in Nature Protocols, and the kind of the checklist and all those kind of technical details are right now available in Nature Protocols as a kind of a resource for those who are interested to have. And I mean, we have multiple Excel sheets and all the tools that you can use if you want to run an fMRI drug cure activity. And there are QR codes if you want to be a part of that collaborative network. So that was the second level. So from networking to coordinated networking, now the next level would be how we can actually cooperate together, how we can share some of the resources that we have in a way to have some compatible goals. We are still working independently to some extent, but we are trying to have some compatible goals together and bring activities together. So the next level would be cooperation, how we can cooperate together. To be able to do that, we started to think how we can ask different labs to share their databases, and then we start to do what we call mega-analysis. So doing analysis in a collection of different kind of large databases that we are collecting from different labs, okay? And we called Enigma mega-analysis. So we started to develop a specific fMRI analysis pipeline for large databases, and we invited all labs that have fMRI drug cure activity data to share their data. So far, we collected over 3,000 participants in terms of the number of fMRI sessions that we collected so far. And we basically put together, now we are kind of putting all those pieces together, and we are running some kind of large data analytics to be able to kind of process these large databases. And it is open, still open for cooperation and collaboration. So if you are interested to share your databases, you are definitely welcome to join that international collaboration. But beyond the mega-analysis, we realized that the tasks that people are using, there is no harmony between what people are doing in different labs for tasks. So we ended up realizing that there might be a good idea to have, at least for those who do not have specific questions about how kind of changes in task parameters would change the brain response, and they want to just have a task that would have a standard outcome, how we can have a task that multiple labs use that task across the board. So now we are working together in a specific task force, and we are running some a series of global surveys asking people, okay, this is the kind of, for example, this is one of the questions. This is the duration of fMRI tasks that people have used around the world during last 10, 20 years. This is the variation that we have. What do you think about what would be the right timing for an fMRI task? So what do you like to have as a kind of the timing of an fMRI task? And there are multiple questions. So we try to provide them with data and what has happened so far, and asking them, okay, what will be the next level if we want to have one single fMRI task that would be used between labs across the board. So that is another ongoing activity that we are doing right now. And then the next level that we are trying to do some cooperation, we started to interact with FDA right now in terms of how we might be able to have fMRI drug reactivity as a kind of FDA qualified biomarker. And the process is pretty complex in terms of how to apply to FDA to make that happen. And before being able to apply for that, we need to have a level of cooperation between different labs to bring pieces of evidence together. To be able to do that, we organized a webinar on October 2020, and we brought people together. And then we had another panel in ACMP 2021 with multiple colleagues, including Nora Volkov and George Koop, discussing about how we might be able to have fMRI-based biomarkers for addiction treatment. And now we are working on what we call an fMRI drug reactivity qualification pathway that we are discussing with FDA and other organizations to be able to make that as a document and kind of apply for the next level. And to be able to do that, the next level would be putting together what we call an FDA letter of interest. But that is a real level of collaboration. Then we have one shared goal across different labs and one specific task. We all want to work together on a specific task to make that happen. This is what we call collaboration. So we started with networking, coordinated activities, some cooperation. Now we are at the level of being able to collaborate and get something done in a collaborative way across different labs to be able to have a specific product. To be able to do that, we are working across different labs to be able to basically collect all the pieces of evidence that we have for fMRI drug reactivity as a biomarker and putting them together to be able to apply for what we call an FDA letter of interest to go for the next level of kind of approval and qualification for fMRI drug reactivity to be used in the field. And going to the next level, the other collaborative activities that we are doing is trying to use fMRI drug reactivity as a targeting tool for neuromodulation studies. Just to give you a really brief overview how we are doing that across different labs, that we already share the databases, the tasks, whatever we have. Now we have the question in terms of, okay, what are the best targets in this Q reactivity network in the brain that we can use to be able to change addictive behaviors with neuromodulation technologies? Okay. We had a webinar July 27, just something like a few weeks ago, discussing about optimized neuromodulation in non-invasive brain stimulation for addiction treatment. You can find that in the QR code. The videos are already recorded, so you can find that. So we had a discussion how we can push that forward. There is a recent publication. I'm not sure how many people that we have in the room, they have heard about this publication in Nature Medicine. This study is, how many others have heard about that? This is a study about what are the brain lesions that could help people or could contribute to when people have the kind of lesion on specific areas in the brain, they basically stop smoking. Those who have been smokers, they stop smoking. Or those who have been drinking alcohol or they had a kind of alcohol use disorders, they basically stop drinking alcohol. And there are in this specific study, I'm not going to go to the details, but they realized there is a specific brain network that if we have lesion on any part of that brain network, that could be a big predictor of quitting smoking or stopping drinking alcohol. And that was a sort of an interesting contribution to the field, because people started to think about, okay, if we can make something like virtual lesions on these areas, or do neuromodulation on those kind of areas, and kind of stop or change their activities, we probably can contribute to addiction treatment as well. So that was a kind of a new publication in the field by a group of kind of colleagues, mainly in Harvard University, but also NIDA and other labs that they have been collaborating together. So we started to think about, okay, when we have fMRI drug reactivity, do we have any shared kind of activations in brain areas that are shared with this specific kind of lesion map that they developed, okay? And these are the activation maps that we have for Q reactivity in alcohol use disorder, smoking, and methamphetamine. And you can see that all of these kind of, and they are from different labs that we are collaborating together right now. They all have shared frontopolar activities that could be targeted, because they are right in the kind of in your frontal area. So we can target them with different neuromodulation technologies to be able to change addictive behavior. So we realize that it's interesting, because it seems that we have converging evidence that Q reactivity would have the same network as what we have in lesion studies. That would be interesting. And now we are expanding that to other databases that we have in Enigma addiction Q reactivity, trying to see how much shared network we have with all those databases that we have. And we realized that the specific frontoparietal area would have connectivities to both amygdala and also nucleus accumbens. So technically, with that frontopolar area, we have something like a window to amygdala and nucleus accumbens to be able to modulate processing, drug Q reactivity processes that are happening on those areas. And that might be a kind of another window of potentials for being able to use neuromodulation for addiction treatment. And we showed that all the previous studies, neuromodulation studies, that they had positive results for addiction. When we do a modeling on the areas that they have been targeting in their studies, they all have a shared area within that kind of lesion-based map. So it is quite interesting that even the studies, for example, these are the TDCS studies, those who know TDCS studies, that they have been putting the electrodes on the dorsolateral prefrontal cortex. At the end of the day, the highest level of electrical current would be in the frontopolar area. So that is the kind of the interesting finding that we have. We are right now kind of submitting a new manuscript around that, and that collaborative network we have. And the other thing that might be interesting is that when we go to the individual level, the map of frontopolar activity in the individual level is quite different across different people. And now we are trying to use that individualized map to define individualized neuromodulation protocols to be able to optimize interventions in the individual level, to be able to have the better results in neuromodulation. So that's another kind of level of activities. I'm not going to discuss about addiction neuromodulation right now, but just to give you an idea about how we can get to the level of what we call collaboration, to have shared goal across different labs, shared activities, and do something together. But to be able to get to that level of collaboration, we had to start from networking. We had to do some coordinated activities. We had to do some cooperation. So to be able to get to that level of collaboration. And it took us something like 4 or 5 years to build up step by step of that level of trust across different labs to be able to get to that level, to be able to do some actual collaboration together. Okay? But that is something that is technically doable. And we are happy to share those experiences with other kind of people who are active in other areas of addiction treatment or addiction prevention, and how that could be used in other areas. As I've mentioned, within the International Society for Addiction Medicine, we have multiple interest groups. I'm one of the co-chairs of neuroscience interest group with Antonio Verdejo Garcia, who was not able to make it for this conference here today. But we use these sort of interest group. And Mark Potenza is the co-chair of behavioral addiction work group. So we have multiple work group or interest group within the International Society for Addiction Medicine. And we try to use the same VCONS model in those collaborative interest group within the International Society for Addiction Medicine. And as I've mentioned, for example, this is one of the kind of initial papers that we had for using neuromodulation for addiction medicine as some like consensus statement. And then, as I have mentioned, this is a new initiative that we started on neuroscience-informed addiction prevention. So that's another level of collaboration that we are pushing forward. And right now we are planning for a global survey around that. And also the recent survey that we have recently kind of finalized about cognitive training and remediation for addiction treatment. So there are multiple activities, but we are open for other collaboration. So if there are people who are interested to develop other collaborative networks, this is what, I mean, this is not an open, this is not a sort of closed community. So it's an open doors community, and we are here to encourage others to basically use this resource and try to see how we might be able to develop more VCONs within the network that we have in the field of addiction. And my last slide, just to make some some take-home notes. How to develop virtual collaborative networks. We usually start with a series of webinars to bring people together, define what are the questions that we have, what are the things that we might be able to do together, kind of define the priorities that we have in the field, making sure there are a group of people who are interested to do some activities. Then we usually do systematic reviews, making sure that we know all the experts in the field. And we try to invite them, making a mailing list of those who are experts in the field, making sure that they are all involved in these activities, whether they are in the US, in Iran, in Egypt, in Australia. So wherever they are, we try to get them involved in the process. And then we start to investigate the parameter space, methodological parameter space within that specific area based on the systematic reviews that we are doing. And we start to encourage people to share their methodological experiences and protocols and develop a level of trust and friendship within the network that we have. And we try to encourage labs to have a level of methodological transparency to help others and share the resources that they have, and also be transparent in terms of the papers that they publish and methodological kind of notes that they publish. And we try to also explore potentials, how we might be able to harmonize some of the activities that we do across different labs. And this is always one of the most complex parts of the activity, because usually scientists are happy to even kind of use other toothbrushes, but not use other methodologies in their labs. But this is kind of the challenges that we have. But when we try to work together and develop those harmonized activities, there are potentials to encourage others to be able to use those harmonized methodological kind of protocols in their labs. And then we try to see how we might be able to find platforms to share databases and do mega-analysis across different labs. Then the next level, we usually try to find some joint funds. If, for example, there are a group of people in Australia that they have a specific kind of opportunity to apply for a specific granting opportunity, we try to support them and we try to use that resource to be able to do some specific activity, or in any potentials that we have in the US to be able to get funding and kind of use that funding across different labs that we have to be able to do some joint activities. One of the major things that we try to do within the network that we have, we try to support junior investigators and those who are from underrepresented minorities across the world. So that is one of the major kind of values that we have in our network, making sure that we can involve others from across the world to be able to be involved in these kind of cutting-edge technologies or cutting-edge research activities. And just not to forget that we have two main values in those networking activities. One is diversity, and the other is sharing, caring, and open science platforms to be able to collaboratively answer big questions that we have in the field of addiction treatment and prevention. I hope that that would be kind of the way that we can tackle the problem of addiction with that collaborative partnership around the world. As Arash and Mark mentioned, we have some resources on ISAM YouTube channel, and we are open to bring others to those sort of collaborative activities. There is a list of ongoing projects that we are discussing with different people, but we are available during the Buffett dinner tonight. So if you have any other ideas or suggestions, we are here to answer questions. One of the other kind of last activities that we are doing right now, we are working to develop a series of courses we are discussing with Coursera and other collaborative, other partners to be able some addiction medicine courses. So that is another kind of activity that we are doing. But the addiction medicine courses that we are planning for, they are not supposed to be just an independent activity. So we are hopeful to be able to use whatever we discussed today in terms of ISAM Global Expert Network, World Addiction Medicine reports, Regional Council of ISAM, and also ISAM textbook to integrate all those activities to reflect between these levels of education and research and global activities to have everybody included in those activities. And we have speakers and instructors, not only from 1, 2, 3 countries, but also from kind of experts across the world. If you are interested to kind of know more about ISAM activities, we have the next ISAM conference in the first week of November in Marrakesh, in Morocco. So you are definitely welcome to join us in ISAM conference in Marrakesh next year. And now we are here to answer questions and discuss about potentials for collaboration. Thank you very much for your attention.